Hello friends, welcome. If you are new to my YouTube channel, then subscribe to my channel and press the bell icon to get early notification when I upload my video on life sciences. वेलकम बैक एंड बैक दी वेलकम। स्वागत है आपका एक और नए वीडियो के साथ। तो चलिए शुरू करते हैं। लेट्स स्टार्ट टुडेज टॉपिक व्हिच इज मेम्ब्रेन ट्रांसपोर्ट। देयर आर टू क्लासेस ऑफ मेम्ब्रेन ट्रांसपोर्ट प्रोटीन्स व्हिच आर ट्रांसपोर्टर्स एंड चैनल्स। Transmembrane movement made by transporters can be either active or passive, but solute flow through channel proteins is always passive. Cells carry out active transport in three main ways with the help of coupled transporter, ATP-driven pumps, and light-driven pumps. As you can see in the diagram, coupled transporters couple the uphill transport of one solute across the membrane to the downhill transport of another. ATP-driven pumps couple uphill transport of solute to the hydrolysis of ATP to ADP and PI. And in light-driven pumps, which are found mainly in bacteria and archaea, couple uphill transport to an input of energy from light, as with bacteriorhodopsin. There are three types of transporter-mediated movement. As you can see in the figure, these are uniport, symport, and antiport. In case of uniport, a single substance moves in a single direction. In case of symport, two substances moves in the same direction. And in case of antiport, two substances move in opposite directions. ATP-driven pumps are often called transport ATPases because they hydrolyze ATP to ADP and inorganic phosphate and use the energy released to pump ions or other solutes across a membrane. There are three classes of ATP-driven pump. These are P-type pump, F-type pump, and ABC transporter. P-type pumps are structurally and functionally related multipass transmembrane proteins. They are called P-type because they phosphorylate themselves during the pumping cycle. This class includes many of the ion pumps that are responsible for setting up and maintaining gradients of sodium, potassium, hydrogen, and calcium across cell membranes. F-type pumps are turbine-like proteins constructed from multiple different subunits. They are differ structurally from P-type ATPases and are found in the plasma membrane of bacteria, the inner membrane of mitochondria, and the thylakoid membrane of chloroplast. They are often called ATP syntheses because they normally work in reverse. Instead of using ATP, they use the hydrogen gradient across the membrane to drive the synthesis of ATP from ADP and inorganic phosphate. On the other hand, ABC transporters primarily pump small molecules across cell membranes, in contrast to P-type and F-type ATPases, which exclusively transport ions. ABC transporters are named because each member contains two highly conserved ATPase domains or ATP binding cassettes. ATP binding leads to dimerization of the two ATP binding domains, and ATP hydrolysis leads to their dissociation. Now, let's know about sodium-potassium pump. As you can see in the figure, the concentration of potassium is typically higher inside cells than outside, whereas the reverse is true for sodium. A sodium-potassium pump or sodium pump found in plasma membrane of virtually all animal cells maintains these concentration differences. The pump operates as an ATP-driven antiporter, actively pumping sodium out of the cell against its steep electrochemical gradient, pumping potassium in, which is also known as a sodium-potassium ATPase. Since the sodium-potassium pump drives 3 positively charged sodium ions out of the cell, for every 2 potassium ions pumped in, it is electrogenic. It drives a net current across the membrane, tending to create electrical potential, the cells inside being negative relative to the outside. Now, let's note down some important notes on membrane transport. Ion channels allow specific inorganic ions, primarily sodium, potassium, calcium, and chlorine. Chlorine channel have 2 subunits and potassium channel have 4 subunits. Calcium pump is responsible for initiating muscle contraction through depolarization of muscle cell membrane. Calcium ion initiate the movement of troponin and tropomyosin on actin filament. This is all about membrane transport. Thank you for watching my video till end.